Thank you, Robert. Maybe a little quick advertisement for the parents in the audience. If you're considering where to send your undergraduate students, Matt gave us a great advertisement. Matt and Dan were undergrads, and maybe at the time were sort of questionable about how they might turn out. That was the genetics bit, okay? The environment was their undergraduate education at K-State, and I think they turned out fine. So
send them to Missouri.
Yeah. That's the trouble with being on the program earlier than some other people. So, well, I appreciate the invitation to share a little bit, and we'll have some overlap. And I think probably for many of you, like me, repetition's good. And so I think as you look through the presentations when you're done, you'll see some common themes among presentations, and I'm certainly going to overlap a little bit what Dr. Weaver said, and certainly some slides that look familiar as I took them directly from Dr. Spangler. But I think that repetition is good because we all say it a little bit different, and we get a little different view on things, and that helps us learn. So you'll get 2 or 3 hits of different concepts as you go through the day, and hopefully that helps you conceptualize those and understand them more thoroughly. My topic today is to talk about genomically enhanced EPDs and selection indexes, and we'll do that with some additional material thrown in sort of as support. And one of the things that I always try and think about is as we work in genetics and genomics, it's pretty complicated. But at the end of the day, we have an objective, and that objective is really pretty simple. That is to help seed stock producers make better and more informed selection decisions. Okay? And we want to bring to bear a variety of selection tools in a palatable and easy to use way for producers to make those kinds of decisions. Okay? And I think you'll all agree so far that these genetic and genomic tools can be fairly complicated. Our goal, particularly on the extension side where Matt and I work a lot, is to try and help you all understand those and adopt those technologies into your selection programs and your breeding programs. And so this is a couple in mid-Missouri that I worked with a bit while I was on faculty at Mizzou. And so they're sort of my poster children. They're the Chryslers. And they were a little bit of a litmus test for me early on about getting ideas across to folks. Great people and registered Angus breeders and really, really pleasurable to work with. But our goal is to help traditional quantitative genetic tools like EPDs and the more recent additions from the genomic technologies. How do we package that in a way that commercial and seed stock producers can use effectively to bring about genetic improvement in their herds? And when I say genetic improvement, I don't mean necessarily making more weaning weight or more yearling weight. I mean making directional change or keeping animals at a certain genetic level in your herd. We don't always have to make them bigger or smaller, or whatever the case is. We can have a target and use these tools to get there. What I'm really interested in, though, is how do I do that precisely? Okay? One of the things Dr. Weaver pointed out very clearly was the influence of environment on phenotype, right? And that clouds our view of the underlying genetic merit of individual animals. Otherwise, we could just go out and look at them and sort them and be done, right? Kind of like coat color. But for many of the traits that are of economic importance to us in beef production, lots of genes at play, and we'll look at a few of those. Environment plays a big role. We talked about heritability and that proportion that's under environmental control versus genetic control. Plays a big role in how much data we need, how much information we need to make some kind of inference about a particular animal's genetic merit. And as we amass information, what we want to try and do is get a better prediction of an individual animal's merit. And what we start off with, and those of you in the audience that are seed stock producers will be familiar with this, when we take and look at maybe making a mating decision between a particular dam and sire, we all typically calculate a pedigree estimate EPD, or just the average of the sire and dam, right? That's called parent average or pedigree estimate. And that's representative of what we expect the average progeny from those matings to look like. Okay? But how many of the progeny actually come out looking like average, particularly average across a whole bunch of traits? all of 0 of them, right? They're always different than what the average is, and part of that's due to differences in accuracies maybe associated with EPDs, particularly the DAM EPD if she's not very well proven. But as we amass information, what we want to try and do is drive this estimate of the progeny's genetic merit for an individual animal closer to its true progeny value, or its true genetic merit, which we never know, except for maybe in cases like median focus where we're at 0.9999 something, right? We never get to 1, but we want to drive our information so that our pedigree estimate EPD, as it amasses accuracy and data, either from individual phenotypes submitted on the animal or progeny phenotypes or genomic information, we want to drive that estimate closer to the true progeny difference for a particular animal. The difference between this true progeny difference and an estimate here of parent average is Mendelian sampling effect, okay? Or said another way, how different is the progeny than the average of the parents, okay? And that's an important concept as we work through some of our genomic stuff, because we take out, in many cases through our deregression process that we'll talk about later, we take apart the EPD and take away the parent average so we get how different this animal is relative to the population from its sampling of genetic merit. One of the things that is important to remember is that matings of animals, recombining sperm and egg to restore diploid genomic status, is a pretty random event, right? The chromosomes that land in each of those individual zygotes is a random event. Which sperm fertilizes an egg is a random event. Lots of induced variation in the populations due to that sampling, okay? We have to try and work around that as we make breeding decisions, and getting more precise information is one way to help do that. So as sort of a signpost here for where we're going to go today, we're going to talk about a number of these components, and I just titled the slide From SNP Chip to Genomic EPDs. And this sort of maps out the chronology of things that happen as we think about developing a genomically enhanced genetic evaluation system for a breed association. And one of the first things, obviously, that has to happen, and Matt and John both talked about this, are assembly of large training or discovery populations that have both available phenotypes or EPDs, as well as genotyping all those animals. That's requisite. If we have either genotypes by themselves or phenotypes by themselves, we're stuck, right? We have to have that stuff overlap. The next step, and we'll talk about this in a little bit of detail, is conduct a training exercise, or in the discovery phase here, called the Genomewide Association study, and there's lots of different ways to do that, to form what we call a prediction equation, or figure out what each contribution of these various SNPs might be to molecular breeding value, and build that prediction equation. The next piece is and a very important one that often we don't talk about a lot. I know Matt mentioned it a little bit, but I think the really important step is validation in a target population, so genotyping some animals that weren't used in discovery and seeing how good our predictions do, right? And that gets to the accuracy of your question a bit. Understanding how reliable these tools are. And I agree with Matt, 5 years ago, I wasn't exactly a champion of this particular technology, but today, it absolutely works, okay? It absolutely works, and we need to get on board. The next step here is once we've got conclusive evidence that the technology is working the way we think it should, is we need to develop some way to incorporate the information from the molecular breeding values that are derived from the genotypes of the animals in these prediction equations developed in the discovery population into the national cattle evaluation. And we'll talk about some ways to do that and why that's important. Then obviously we have to have producers that test animals, right? So this is a commercialization piece. Once we have the tools in place to calculate MBVs and incorporate that data, it's time for producers to get busy and genotype animals to build information into those animals' EPDs via genomically enhancing them, and subsequently the selection indexes that come out of those prediction systems. So you can see it's a pretty involved process. There's lots of steps, lots of players, and it takes a fair bit of time and money, blood, sweat, and tears to make all of this happen. But at the end of the day, what we we come out with typically is a methodology that results in producers having a more reliable tool to use in selection that is on the same base and scale and terminology as the EPDs they're already familiar with using. And that's an important outcome, I think, of making genomically enhanced EPDs. We'll talk about some other benefits as well. One of the important things to recognize, and Matt, I think, alluded to this in his talk a bit, is there are certain times when maybe genomic tools might be more useful to us than others. And certainly we started off with weight traits and some carcass trait work because those happen to be the phenotypes we had at hand. But in reality, what we really want to go after are traits that are more difficult to measure, more expensive to measure on a routine basis, because then we can apply and spread those development costs across a larger population of animals when we go to testing, right? If we think about how many of y'all are willing to go out and do a disease susceptibility trial for us to help build genomic tests. That's what I thought, right? So we can test and focus our resources on a small group of animals, build a tool, and then deploy it across all of you. If I ask you how many of you would want to test for genetic resistance to disease, say at $5 an animal, everybody should raise your hand, right? Okay. So there's different times when markers are helpful. One of the most important things is as we develop the tools is having a good understanding of how much of the variation, how much genetic variation that is, a particular DNA marker panel accounts for. And that gets at this association between what our expectation is between the MBVs, the molecular breeding values that we produce, and the EPDs that may already exist on those animals. Okay? Again, we don't expect that when we have a panel that say only accounts for maybe a third of the genetic variation. We don't expect those to line up exactly with the EPDs because it's a part-whole relationship. And we'll talk about that in a little more detail as we go ahead. The other obvious thing is that as we do genotyping for quantitative traits, we get an abundance of other information that's useful to us, particularly from a pedigree livestock perspective. And one of those is that we can do a better job evaluating and reinforcing pedigree structure in our population, right? Because we can immediately, on any animal that's a progeny of an AI sire, we can test for parentage, okay? Make sure the pedigree sorts out the way we think it should. That's something we should do on a routine basis. I'm not sure how many breeds have actually started to do that yet, but it can be a pretty important component because as you look through breed association databases and go back and genotype the pedigrees behind them, somewhere between 5 and 7% error rate in pedigrees, okay? Does that let us do a good job in estimating heritabilities or any other parameter that's derived from pedigree structure? No, it induces error. And so several organizations, I know American Hereford is already going to a system where if you turn out a bull on cows and expect to register calves, he has to be genotyped, okay? And parent verified, okay? And that's to enforce pedigree structure, okay? Pretty good tool. Some traits that are obviously likely to benefit, this is from a colleague, Allison Van Eenam at Davis. Things that are simply inherited genetic defects, obviously one that this tool is very keen to work on and solve and help manage. Carcass quality, palatability attributes, particularly palatability. We don't collect very many of those phenotypes in our business, and so deriving that information and driving it across the sector is useful. Fertility and reproductive efficiency. Notice at the bottom of the list, growth performance. One of the things that I hope will be said again and again and again today is that genotyping is not a substitute for phenotyping, okay? We need to continue to collect performance records on animals across the population, across almost all the traits, okay? This shouldn't be an excuse to not collect birth weights or weaning weights or yearling weights or carcass data or any other phenotype that we can readily get our hands on, okay? It's not a substitute for that. And when we think about refining these tools, the likelihood of us needing more phenotypes is probably greater than needing fewer phenotypes, okay? So, when we think about, and Matt, thanks for the slide, discovering these marker gene effects, so there's a little bit of difference depending on who you talk to about terminology, and I kind of use them interchangeably. If you hear somebody say discovery population or training population, it really means the same thing, okay? And it's this idea of building a group of animals that are phenotyped for the traits of interest, and also genotyping those animals to be used in this discovery phase or the early part of our work to discover the associations between markers and the resulting either genotype or EPDs or phenotypes. Okay? One of the methods we use to do that is a system called Genomewide Association studies. And so what we do is, instead of looking at individual markers or individual candidate genes in the population for variation with a specific trait, we try and do them all at once. Okay? And there's a variety of methodologies to do that. I'm not going to bore you with the details on any of them because I don't think it's particularly important that you know. And it's pretty statistically involved. But what our goal is, and I'll illustrate this in a simple way in just a minute, is to find the relationship between an individual marker and a phenotype or EPD. Okay? That's all we're doing. Okay? And we want to do those across the whole genome. And of course, the more SNPs we have to a certain extent help us with that, particularly as we move from maybe a few hundred SNPs to a few thousand SNPs. We do a better job saturating the genome and finding instances based on differences in marker frequency in the population. We have better chances or less chances of discovering association depending on what structure we use in those designs. But our goal then is to produce molecular breeding values which describe a subset of the genetic variation of a particular animal for a particular trait. Okay? It's not all the genetic variation, it's only the stuff we can find. Okay? The markers we use are markers called SNPs, and it's just a single nucleotide polymorphism. So this is 2 pieces of genomic sequence from chromosome 6 in cattle. Here's chromosome 1, there's 2 chains here, and then the second chromosome. And they're all the same except for this spot I've illustrated or highlighted in the green box. And this is a GT substitution. So it's GC on this strand, and the subsequent TA, we used to only score the top strand. It's a TA in the second one, that'd be a GT. And what we know is that in many cases, the mutations that we score here were discovered and exist largely in the introns between genes, so the non-coding sequence. Okay? One thing that would help us, and Dr. Spangler referenced sequencing that's going on, is identification of causal mutations, or these kinds of SNP changes actually in the coding sequence of a gene. That would be really useful, but not the state we're at today. So this technology we use today, these genome-wide association studies, are illustrated here. This is a single gene example, or a single locus example. Here we have a marker that's a G and T, a G on one chromosome of this bull, and his alternate allele is scored at T. And we know that these 2 markers happen to be, through our discovery process, relatively close and associated with a favorable allele and an unfavorable allele in a consistent way across the population that we've scored. Okay? So when we see a G, it's most often associated with favorable performance. Let's say this is weaning weight. And when we see animals that have T's, either homozygotes or heterozygotes, they tend to have less favorable performance. Okay? So all we do is use the markers as sort of, you can think about them as ear tags, just to track a piece of genomic material through the population and its expected association with favorable or unfavorable forms of a particular gene. Okay? That's all we're doing. Okay? We just happen to do a whole bunch of them at the same time for an individual trait. So, what does an individual DNA marker tell us then? It tells us about the simple association between that marker and some nearby QTL, okay, that's associated with a trait. That's it. Okay? But what do we know about most traits of economic importance in beef cattle? Are they affected by one gene or a whole bunch of genes? Okay? A whole bunch of genes. And that's a little bit, I think, Jerry, early on we'd hoped that there were large single gene effects for a lot of traits, or at least a few genes with very large effects. The downer is that there's lots of genes with very small effects, and that makes them very difficult to find. But we need to score lots and lots of markers and discover these associations to gather up that amount of genetic variation that we desire from the population. I should say one other thing. When we do that, let's say this represents all of the genes with known effects for weaning weight. We have an estimate of the favorable and unfavorable forms of each of those markers. we can add up the net effect of all of those genes to calculate what we call molecular breeding value. Okay, it's just a summation of all of those favorable and unfavorable effects across an individual animal's genome for a trait. That sounds remarkably familiar with something else that you already use every day, okay, called EPDs. An EPD is the sum of all of the genetic effects and interactions between genes for a particular trait, okay? So this gets at the idea that there's a part-whole relationship between a molecular breeding value and an EPD, okay? Those things overlap. They're not exactly concordant because we don't have markers on all of the genes that have an effect on, say, weaning weight. That's represented by the black box around this, okay? So when we take and decide to make a genomically enhanced EPD, what we do is we gather up all the sources of information that account for variation in a particular trait and put them together, okay? That's what this does. And we'll talk about a little bit how to do that as we move ahead. One of the things that's really, really important to understand is that there's lots of variation, and the variation is not the same across breeds. This illustrates some work out of Dr. Taylor's lab. I'm pretty sure this was Matt McClure's work, using SNP50 data generated off of animals involved in the NCBA Carcass Merit Project for Warner Bratzler shear force. This panel here in the lower left is Charlet, Angus, Hereford, and Simmental, and it represents each marker then as a dot on here, and it represents that particular marker's influence or association, allele substitution effect in this case, with Warner Bratzler shear force. Okay, what you can see is there's a whole bunch of them, and lots of them have very small effects. Okay, one of the challenges in adopting genomic technology was early on there was two pieces of information that floated around out in the business. Okay. And still, in some cases, there's the same challenge that exists. One of those, obviously, is the EPDs that are published regularly on animals, and again, it's the sum of the additive effects of all genes that influence a given trait. The underlying genes are unknown. We calculate these a bit blindly that way. We don't know what the underlying genes are, just the net effect of them per animal. And we have some challenges associated with the time delay of collecting records. Think about maternal calving ease or stayability as two examples. It takes a long time to collect that information because we have to have daughters in production. The other piece of information that's generally available is molecular breeding values, and they are the sum of the allele substitution or SNP effects, and that is again a sort of a part-whole relationship between the EPD. They're not the genes, but they're associated with genetic variants associated with the trait. One advantage of these, obviously, is they can be collected at or near birth, right? So we can get information about traits that may be expressed much later in an animal's life, very early in their life, and use that information in selection. The key thing here, these are not substitute goods, okay? We have to use them together. We have to figure out how to use them together. Well, the way we do that, in a simple way, is we incorporate information from the molecular breeding value and the EPD to calculate these genomically enhanced EPDs. And there's a variety of ways to do that. 4 general ways that are being used today in the beef industry. American Angus, a number of years ago, deployed a methodology that fits the molecular breeding values as a correlated trait, so that they actually get a genetic relationship in their genetic evaluation for that data. It's very similar to how we use ultrasound data to inform carcass EPDs. We know there's a genetic association between ultrasound records and carcass records. We model that association in the genetic evaluation for carcass data. We can do that same sort of thing and do that same sort of thing for molecular breeding values. Another methodology is a blending method, basically where we create, in many ways, you can think of it as a selection index, where we're blending 2 pieces of information and we weight them based on accuracy, right? So if we have a high accuracy bull that has an EPD and we provide molecular breeding value information, it has very little effect on the subsequent genomically enhanced EPD because of the disparity between accuracies of those 2 measures. But if we have a non-parent animal that, say, maybe only has their own phenotype recorded, has low accuracy EPDs, has genomic information provided, those are much more on kind of the same footing, so that provides a boost in accuracy for those particular animals. This method is used by American Hereford and soon to be used by North American Limousine Foundation. Treating as an external EPD is a method that is used at American Simmental. They, for a long time, as a function of their multi-breed genetic evaluation, have leveraged data from other genetic evaluations to properly rank bulls represented from other breeds outside Simmental. In many ways, this genomic information can be viewed exactly the same way. Okay? It's external data about the genetic merit of an individual animal relative to other animals. Okay? And so they have a method to pull that data in to their evaluation using that architecture. And then lastly, one that hasn't been used much in the beef business, but used routinely in the dairy business, is the development of the genomic relationship matrix and using actual genotypes directly in the evaluation in a single-step process. A little more computationally intensive, but has been fairly successful for the dairy business. A little bit different paradigm in the way their systems are structured versus ours. But some work obviously been done in beef. In fact, Jerry Taylor and I had a student who did a lot of work on genomic relationship matrix. So these are the kind of the 4 different methods. One of the key things to represent or know about them is none of them are perfect. Okay? There are things that we would like to improve about each of these methodologies. But the reality is they all do a fairly good job of taking the information and turning it back into a tool that you can reliably use as producers to make selection decisions. And I think the most important contribution the genomic tools make to your selection system is through improvement in accuracy. Okay? One of the things that I routinely hear from breeders, seed stock producers that is, that genotype animals is, well, shoot, my EPDs went down. Was that expected or not expected? It's expected, right? In some cases, the additional information we gather from the genotyping says this animal got a pretty lousy assortment of genes, and the EPD really just hasn't had time to catch up yet. Okay? And vice versa. We can have animals that maybe have mediocre EPDs but happen to have a very good sampling of genetic material that's been evaluated by the genomic test and actually move the EPDs up. So we shouldn't make an expectation about animals' EPD directional change. What we should make an expectation of is that the tool that we get back, the EPD we get back from a genomically enhanced one, is a better predictor. It's more reliable, has higher accuracy. And that's our objective, right? If we go back to the very beginning, the very first slide I showed you with the Chryslers, we want to provide a very useful and reliable tool for people to use. Okay? That means more information used to calculate the metric. Okay? The increase in accuracy is proportional to the percent of genetic variation that the DNA marker panel explains. Okay? We have a panel that explains lots of the genetic variation in a trait. The boost in accuracy we expect to be fairly high. If the marker panel only accounts for a small fraction of the genetic variation, genotyping animals, providing that data to the NCE system, will result in a very low increase in accuracy. Okay? You probably have all seen a slide like this before, and it just illustrates the importance of accuracy. Okay? Here's two bulls. Let's use weaning weight as an example. Let's say it's a 40 weaning weight EPD. 2 bulls, exactly the same EPD, dramatically different accuracy values, a 0.3 and a 0.8. So the 0.3, this would be representative typically of a yearling bull you might have in your spring or fall production sale, available for clients to buy. The one that's a 0.8 would be what? An AI side, right? Fairly well proven. We don't expect his EPDs to move very much. The amount we expect his EPDs to move is represented by the possible change or standard errors or predictions associated with these 2 bulls' different accuracy levels. What you notice about the bull in red, he has a much wider distribution that his EPD might change over time as more data is collected than the bull in blue, right? Which would you rather sell to customers? The one in red or the one in blue? Let's think calving weeks, okay? Do you want the phone calls or not, okay? It's a really important contribution to genomic technology. We're not going to move them all to 0.8 today. Hopefully one day we can move them to 0.8. That'd be fantastic. If you could all sell yearling bulls with EPD accuracies of 0.8, Jerry and John would be bigger rock stars than they already are, okay? Be really useful. But the idea is to get more accurate prediction because we are more confident in the product we sell. that accuracy is represented. I'm a lefty. I should have thought of this a long time ago. I like pictures of things, particularly things associated with statistics, because it helps me get them. This is a picture of differences in variance. Okay, so this is the green out here represents all of the phenotypic variance. If you remember from Dr. Beaver's talk, phenotypic variance is contributed to by what? Genetics and environment, right? So the white circle here represents genetic variation, and then the whole circle here represents phenotypic variation. So the green part in between represents environmental variation. Okay, so this trait has a heritability of 0.4 or 40%. So this white circle has 40% of the area. The green represents 60% of the area. Okay, so everybody got that? Okay. There's a DNA marker test that accounts for a third of the genetic variation. Okay, so this blue represents a third of the white space. Okay, everybody good? So what proportion of the genetic value or phenotypic variation is accounted for then by the DNA marker panel? Okay, somewhere around 12.5%. It's the product of the heritability and this percent genetic value. Okay. But what do we know then if we have a panel that describes a large chunk of the genetic variation? In this case, 80% of the genetic variation. It soaks up almost all but 20% of the genetic variation in the trait. We describe much more of the phenotypic variation as well. Okay? What we'd expect is if we calculated the contribution to accuracy from this particular marker panel versus the previous one, this one would do a lot better job because it explains more of the genetic variation in that particular trait. Okay? And this goes to the earlier question about why we don't expect the EPDs and the molecular breeding values to have strong correlations. If we had a panel that accounted for 80% of the genetic variation, we'd expect a very strong relationship between an animal's MBV and its EPD, because they're almost identical in terms of the space they occupy. Okay? But in the previous slide, 2/3 of the genetic variation was unaccounted for. So our expectation of the resulting correlations between those two is much, much lower. Okay? Impact on accuracy, Dr. Spangler already showed this slide. Almost all of the benefit accrues to animals that are low in accuracy. Many of you at first blush might go, oh, crap, that's no good. Well, in fact, it's actually exactly what we want to do, right? It's exactly what we want to do, and it impacts the population that we need to impact. Okay? We want to build accuracy on animals, reliability on their EPDs as early as we can in their life to make selection decisions, right? If we capture information about an animal's genetic merit after a selection decision has occurred, does that information do us any good at all? The only good it might do us is tell us how many phone calls we're going to get from dissatisfied commercial producers. Okay? That's the only potential benefit. But wouldn't it have been much better to actually make those selection decisions earlier in the animal's life before you had that opportunity? Okay? So accruing this accuracy early in life is our objective. I want to talk for just a minute. Selection index was in my title, and I've pretty much ignored it up to this point. Because the benefit that genomic testing brings to selection index is directly through the improved accuracy of the EPDs used to calculate the index. That's the only contribution I can see that genomics provides to selection index. But that's not to say that it's trivial. Okay? Selection index, I think, is a very important tool for commercial producers to use, and you guys are one of the pioneers in developing your terminal sire index. And using EPDs that are higher accuracy in producing those indexes makes the index of higher accuracy and better reliability, and very, very important to producers. And I hope as things continue, particularly as it relates to our feed efficiency project, that more indexes are generated, particularly ones that incorporate information from the cost side of our business represented by feed intake and feed efficiency. Okay. One of the last couple of slides here I want to conclude with is sort of giving you an idea, if you're not already familiar with this sort of flow of information, is how, as a producer, this system might work. As a producer, you collect a DNA sample, and in many cases, you still need to submit that through your breed association. They forward that information then to and the sample with some identification information to a genotyping service provider. That company genotypes the animal on what the respective technology platform that they use to calculate or produce genotypes, subsequently use that information to compute molecular breeding values, which is then reported back to the Breed Association, incorporated into the National Cattle Evaluation, that's what NCE stands for here, and either marker-assisted or genomically enhanced, same thing, EPDs that get returned back to producers. So that's the kind of the cycle of information that you should expect. And I would encourage you, in some cases, you may get the molecular breeding values back directly from your service provider. For many of you, I don't think that'll provide any meaningful information to you aside from getting it stuffed into your EPD. Okay? Dissecting the difference between an animal's MBV and its EPD and trying to understand why those two are different is, well, it's not a trivial exercise, and I don't think it's one that you'll be able to come to any good conclusions doing. Okay? You might be burning computer cycles that you don't need to burn. Rely on the genomically enhanced EPD to help inform those selection decisions. I'm going to skip a couple of slides here because Matt covered this stuff fairly well about inclusion of the data. I want to stop with this slide, and I think it's a really important concept. How many of y'all in the audience are seed stock producers? Okay, I thought almost everybody here would be. If I had a soapbox to stand on, I would stand on it. Okay, this is really important. If you don't take anything away from my talk this afternoon, take this. Okay, there's a big difference between marker-assisted marketing and marker-assisted selection. Okay. Big difference between marker-assisted marketing, not marker-assisted management, marker-assisted marketing, marker-assisted selection. If you only use the genomic tools to genotype maybe the front 5 or 10 bulls in your cell catalog, try and market them to an AI company, you're doing marker-assisted marketing. Okay? You haven't done anything to inform your selection decisions or change the genotypes of the bulls that are going out into the commercial industry. Okay? To do that, you actually have to use the tool in selection, which means getting the animals genotyped. Probably almost all of the animals in your calf crop are a contemporary group. You can knock off the obvious outliers in terms of, horn poll or coat color or, the 100-pound birth weights or the C-sex. Any of those ones that you're not going to sell no matter what, throw those out of your genotyping exercise. Genotype everybody else, okay, if you're going to do it. And then use that information to make your selection decision on which bulls go into the development pen. Now you've changed, or potentially changed, gene frequency that's flowing out of your operation. Okay? The other way, okay, if I use marker-assisted marketing, genotype the front 5 bulls, all the bulls that you go and sell are going out to make cows somewhere, and likely all of them are going to make commercial cows. We haven't done anything to change gene flow in the population. Okay? So there's a really important context to think about as, I know all of you have a business and you sell a product. Okay? I understand that. Okay? But if we think about the Charolais breed making genetic improvement, using this technology to make genetic improvement means that you have to engage in marker-assisted selection. Okay? You have to be able to discover these new animals that have unique combinations of genes in a desirable format to make a gene flow change. You have to do that collectively. Okay? Step off my soapbox. With that, I'll stop and be glad to answer any questions. Appreciate y'all's attention.